Hello, everyone. It's really my pleasure to share with you my studies on the blood-based biomarkers of Alzheimer's disease, or AD. Let's talk about the dementia first. Dementia is a general term for loss of memory and other thinking abilities severe enough to interfere with your daily life. It includes majorly Alzheimer's disease and also other neurodegenerative diseases. So the general symptoms include the memory loss, as we know, difficulties in performing familiar tasks, disorientation to time and place, poor or decreased judgment, changes in mood, personality, or behavior, problems with images or spatial awareness, misplacing things, withdrawal from work or social activities, problems in language and communication, difficulty planning or solving problems. So now it becomes a global crisis. Currently, there's around maybe 50 million patients worldwide, and most of the cases are in Asia. So the prevalence is around 1% worldwide, with higher rates in the elderly and more in female. It is estimated that it will double in every 20 years. Now let's talk about Alzheimer's disease. Compared to the normal brain, there are shrunken hippocampus and cerebral cortex, and also enlarged ventricles in the AD brain. Let's take a closer look at the cellular level. So we can see that there are extracellular amyloid plaque and then intracellular tau tangles. With the disease progression, there are a few AD biomarkers to recognize. The first is amyloid beta, and then tau protein in cerebrospinal fluid, and then the brain structural change, memory loss, and finally, more clinical function loss. So it seems likely amyloid beta and the tau proteins are the initial biomarkers or the steps in the pathogenesis. However, these methods are either too late to know or that's too difficult to do. So currently, the physicians will majorly use neuropsychological assessments, including the one majorly used is mini mental state examination or other assessments, and also some blood tests, mainly for rule out other possible diseases. And nowadays, the neuroimaging is more important, including the CT or MRI. But these are not very specific for diagnosing Alzheimer's disease. Okay, so let's look at the pathological cascades of AD. So it starts from this protein. It's a membrane protein. We call it amyloid precursor protein, or APP. So normally it is processed by secretases, but when it is processed by beta and gamma secretases, it generates a beta, especially 42 with 42 amino acid residues. It's more toxic than other isoforms. It will misfold and form the oligomers and then form the amyloid plaques. That's what we see in the extracellular area in the brain. Okay, so the A beta oligomers and the plaques itself can damage the neuron, especially at the synapse region. Also, it generates oxidative stress, which can affect a certain group of kinase activities. Then the hyperphosphorylate tau protein, which is a stabilizing protein for microtubules. So the hyperphosphorylated tau will misfold and form neurofibrillary tangles inside the cells. So both the amyloid plaque and tau tangles can damage the neurons. Although most of the AD cases are sporadic, but still some genetic basis. There have been found a few genes mutated in the family of AD. For example, the APP is a membrane protein associated with kinesin, a microtubule-dependent motor protein, and also the gene for presenilin is an APP cleaving enzyme, tau protein, and microtubule stabilizing protein, as we said. And ApoE is a lipid shuttle in low-density lipoprotein. Been reported that in the familial AD mutants is a mouse model with the mutations in presenilin 1 APP genes. So you can see the transport of either the LDL or APP or other vesicles. It's been disrupted in the disease neurons. As you can see, the distribution of these markers were stopped in the cell body and the fewer in the neurites. So in our research model of the vesicle transport biomarkers in the microtubule-dependent vesicle transport machinery containing the motor proteins and adapters is vital for neuronal survival by delivering neurotrophic cargoes, including the motor proteins, kinesin, dynein, dynectin, adapters, including Huntington protein, Huntington associated protein 1 or HAP1, or Belson helper integration site 1, AHI1, and other neurotrophic cargoes, including the APP, the track A, B, the BDNF, the BCR, ApoA1, and more. So all these proteins and their modified products could be potential biomarkers for the disease. So we tested them in human blood. So using Western blotting, we did a quick screening on these potential proteins, including AHI1, Huntington protein, kinesin, light chain, HAP1, and BCR, APP, and ApoA1. With the statistical analysis, we found the significant reduction of AHI1, Huntington protein, kinesin light chain 2, and APP in the AD blood. And then we collect more human samples to test and do ELISA, and found that the significant reduction is found for AHI1 in the AD serum, but not the others, including the amyloid beta, which seems a good marker in cerebral spinal fluid, but not in the blood, actually. The Huntington protein and kinesin light chain, it seems not as good as AHI1 as the marker for the disease. So in the disease model cells in the mouse brain, we also see a reduction of AHI1. As we can see in the disease model expressing the mutant APP, comparing to the wild-type APP, you can see the reduction of AHI1 with a statistical significance in the cell model. Similarly, in mouse brain, including the cortex, the hippocampus, and also other regions, you can see the AHI1 also reduced with a statistical significance, the AD mouse brain. Okay, so to check the diagnostic performance of AHI1 for AD, so we did receiver operating characteristic analysis, which determined the sensitivity or the true positive rate against the false positive rate. So then calculate the area under the curve. So with all about 86 samples, so we got the area under the curve of AUC around 0.7, which is fair. So then we checked the concentration of AHI1 against age and found that for health control, you can see the concentration of AHI1 decreased with age, but the concentration in Alzheimer's disease patients stay about the same. So it seems likely it's a better marker in the early stage of the disease. So indeed, when we look at the ROC analysis, and we found the area under curve is increased to about 0.82 with the younger subjects. So it turns out that AHI1 could be a good early marker for the disease. So in summary, AHI1 is reduced in AD model cells, mouse brain, and also AD patient serum. The concentration of AHI1 in the control serum was decreased in an age-dependent manner, while it was constantly low in AD serum. The receiver operating characteristic analysis suggests that AHI1 is a good diagnostic marker for the AD, especially at relatively young age. So the serum AHI1 may be valuable for early diagnosis of disease. However, larger sample size may be required. So this has been published in 2 papers in the past 2 years. One is on the mechanism how AHI1 expression can rescue the pathology of the AD model cells, and the other is to evaluate the AHI1 as a biomarker in the blood of AD patients. So when we look at the clinical chemistry data, we found there is a significant difference between Alzheimer's disease group and the health control. That is fasting glucose. You can see that is significantly higher in AD group. So it is also known that some diabetic patients will develop Alzheimer's disease. But the question is how? So in the blood, there are certain kind of glycation end products that is derived from glucose. Okay, so the glucose in the blood will either react with protein first and go through some kind of oxidation, fragmentation, and then form the carboxymethyl lysine, or during the glycolysis, the byproduct such as methylglyoxal will react with proteins and form carboxyethyl lysine. Of them are called advanced glycation end products, or AGEs. It also been proposed that AGEs and their receptors are also responsible for Alzheimer's disease pathogenesis. Let's see how we test it. So first, we get a serum protein, which is abundant and has no big difference in the blood. So we choose apolipoprotein A1 or ApoA1 as an AGE carrier. So we run a gel with the pooled serum from either control or Alzheimer's disease patients And then we cut the gel and did the mass analysis. Fortunately, we really found CEL modified ApoA1 peptides, especially in AD patients. Okay, so to verify, we test the serum with CEL antibody, and then we found there's a significant increase of CEL adults in AD serum. Okay, so we use ApoA1 antibody to precipitate ApoA1 and then do the blotting using the CEL antibody, and we found that ApoA1 has CEL modification. CEL is really more in AD serum than in control serum. So indeed, the advanced glycation end products CL is significantly present in AD serum. It's also proposed autoimmunity contribute to the AD pathogenesis. So then we check the specific IgM and IgG for the CEL and found that IgM against CEL is significantly elevated in AD serum. It gave us a very good diagnostic performance with the area under the RC curve around 0.95, which is very good. So it's related to the disease progression. So we plot the concentration of either IgM or IgG with mini-mental state examination score. So the lower score is more severe in the disease. We see there's a positive correlation. It means that the autoantibody IgM is elevated in early AD stage and then decrease with AD progression. So in summary, the advanced glycation end product CL is significantly increased in AD serum. So the anti-CL autoantibody, especially IgM, is elevated in early AD and decreased with AD progression. So CL and responding autoantibodies may be good biomarkers for not only diagnosis but also immunotherapy for AD patients, especially those with diabetes. So it's also been published. Right now, we still have a project working on the biomarkers in the pathogenesis from hyperglycemia to neurodegeneration. Now we found that the hyperglycemia can increase the level of HNE and A beta through oxidative stress and lipid peroxidation. And both HNE and A beta can be neutralized by responding autoantibodies. When these autoantibodies are depleted, the patients may develop neurodegeneration, such as AD. So the HNE and responding autoantibodies also good biomarkers for diagnosis and could be also immunotherapy for AD patients with diabetes. So at the end, I'd like to thank my lab members. Although most of them have graduated, but we still have someone still working on this project. And also like to thank my colleagues in Taipei Medical University and the hospital, and also friends in Chang'an University of Science and Technology. Thank you very much.